Hello everyone. Today we're exploring the fascinating world of genetic engineering, what it is, how it works, and the ethical questions it raises. We'll look at how scientists modify DNA, the tools they use, and the way this technology is applied in medicine, agriculture, and research. Genetic engineering is the process of intentionally altering an organism's DNA to produce desired traits. Scientists can add new genes, remove harmful ones, or modify specific sequences to achieve precise outcomes. Unlike traditional breeding methods, which rely on random inheritance over generations, genetic engineering allows for targeted, controlled changes in plants, animals, and microorganisms. Before gene editing technologies, humans relied on selective breeding, where individuals with preferred traits are mated and the offspring inherit a combination of genes. This method is time consuming, unpredictable, and can unintentionally maintain undesirable traits. Genetic engineering, by contrast, enables scientists to directly manipulate specific DNA sequences. This precision allows for faster, more reliable outcomes and can introduce traits that would not occur naturally. Genetic engineering has a wide range of applications. In agriculture, it allows crops to resist diseases, pests, or environmental stressors, and can enhance nutritional content. In medicine, bacteria or yeast can be engineered to produce insulin and other therapeutic proteins. Gene therapy can treat genetic disorders, and transgenic animals provide models for research or potential organ transplantation. These applications demonstrate how genetic engineering can directly benefit society. One of the most significant advances in gene editing is CRISPR-Cas9. The guide RNA acts like a GPS, directing the Cas9 enzyme to a specific sequence in the genome. Cas9 then makes a precise cut in the DNA. The cell naturally repairs this cut, and scientists can take advantage of this repair process to add, remove, or modify genes. CRISPR is precise, efficient, and has revolutionized modern genetic research. Beyond CRISPR, there are several important genetic engineering techniques. Agrobacterium transfer uses a bacterium to deliver DNA into plants. Microprojectile bombardment shoots DNA-coated particles directly into cells. DNA cloning allows for scientists to replicate specific genes for study or insertion. Genetic edits can be made in somatic cells, affecting only the individual, or germline cells, which can be inherited by future generations. Genetic engineering has transformative potential in medicine. Gene therapy can correct defective genes to treat inherited disorders. Immune cells can be edited to more effectively target cancer. Engineered organisms produce vital pharmaceuticals, including hormones and clotting factors. While embryo editing remains highly regulated, research continues to explore its potential for preventing genetic diseases before birth. Genetic engineering also presents significant ethical considerations. Editing embryos could prevent diseases, but may also lead to the creation of so-called designer babies. Access to these technologies is uneven, which could exaggerate social inequalities. Ecological concerns include the potential impact of genetically modified organisms on natural ecosystems. These issues highlight the need for thoughtful regulation and public dialogue. When evaluating genetic engineering, it is essential to weigh benefits against risks. Long-term consequences, societal impact, and environmental effects must all be considered. Transparency in scientific research and inclusive public engagement are critical. Ethical guidelines should ensure respect for human rights and prevent misuse of these powerful technologies. In summary, genetic engineering provides unprecedented control over the DNA of living organisms. It holds immense promise for improving food security, treating disease, and advancing scientific knowledge. However, with this power comes responsibility. Careful ethical consideration and regulation are essential to ensure that these technologies are used safely and equitably.